Today we're going to talk about enzymes. So I have up here this little cartoon that says what the enzyme told his substrate. So this little pink guy is the substrate molecule and the blue guy is the enzyme. So what the enzyme told his substrate, you complete me. So if you look at the two of them, right, they look like they're kind of almost, I don't want to say mirror images of each other, but the fact that this guy will fit into the opening on this one, right? So the shape of that substrate mimics the shape on the enzyme there. And so that's why this guy can kind of fit in here and it will complete the enzyme. So let's talk about this process and what does all this mean? So enzymes, we say, are catalytic proteins. So they're catalysts, which means that they help to speed things up. And what they speed up are chemical reactions. All right, so enzymes are types of proteins. So we've talked about proteins before. We know that proteins are very complex molecules and that the shape of a protein is very important. So in biology, we always say that structure determines function. So the structure of the molecule is very important for how it functions. And proteins, we know, can fold into all different kinds of shapes. They're taking their amino acid chains and we're folding them into different shapes so that they all can perform different functions. And enzymes are just one type of protein. And these are proteins that help to speed up chemical reactions. So we say they change the rate of the reaction. So let's say I have a glass of water here. All right, so I'm going to draw something out. And here's my water level. And I'm going to place a sugar cube in that water. Now, if I heat up the water, that means the water molecules are going to start bouncing around because they'll have a lot of thermal energy. So all these water molecules will bounce around. They'll be bouncing into the sugar molecule, and that will help to dissolve the sugar cube. If I place a spoon in here and I stir the water, once again, I'm adding energy to this system, and the water molecules will be bouncing around and they'll start bouncing into the sugar cube, and that will help to dissolve the sugar cube. But what if I don't do anything? What if I don't add any energy to this system and I just allow that water molecule, the sugar cube to sit in this little glass of water? Will that sugar cube dissolve? Sure, it'll dissolve. Might take a really long time, but it will dissolve. What if I add an enzyme to this watery solution here? If I add an enzyme like sucrase, then that enzyme helps to change the rate of the reaction. So it helps to speed up the reaction, helps to catalyze the reaction. So therefore that sugar cube is going to dissolve much more quickly than if I just left it on its own. So if I add energy to the system, then yeah, I can get that reaction to proceed. But without it, if I don't add energy, I can still add an enzyme, and that enzyme helps to lower how much energy is needed for that reaction to proceed. And so therefore it changes the rate of the reaction. It can proceed much more quickly if I add an enzyme. All right, so we say that enzymes are named after the reaction they catalyze. So they really kind of give you a clue as to what their job is, since they're named after the reactions. And we also tend to find that they end in the ASE. So here we have the example of polymerase. It has that ASE ending right there that lets you know it's an enzyme. And we've got the word polymer. So this is an enzyme that makes a polymer. So it could be DNA polymerase, which helps to make the DNA molecule, or it could be RNA polymerase that helps to make RNA. So it's named after what they do. I have the example down here of carbon dioxide and ribulose biphosphate. These are our reactant molecules, and they are going to be converted to the product of phosphoglycerate. In enzymatic reactions, we call our reactant molecules substrates. Okay, so instead of reactants and saying these are going to react together, we say these are the substrates. And the enzyme that helps to catalyze this reaction is called ribulose biphosphate carboxylase. So that's a mouthful. But notice it has this ASE ending on it, and it's named after the molecules that it works on. So we've got the ribulose biphosphate, which we had up here as one of our substrate molecules. And it's a carboxylase is kind of what it's going to do. It's going to work on ribulose biphosphate, and it's going to carboxylate it. So this is kind of a little bit of an intro to the enzymes, how we name them, and what their overall function is. So let's look at how this works. So over here we have an enzyme. So these kind of orangish, dimply looking structures are the enzyme. So it's representing, like, each one of these little circles kind of represents an amino acid that's making up the structure of that enzyme, since it's a protein. And we notice that there's a hole right here. All right, there's a portion of the enzyme where the substrate molecule is going to come in and bind to the enzyme. And this area is called the active site. So it's only a region of the enzyme, the overall enzyme, in which the substrates bind to. And the shape of that active site typically mimics the shape of our substrates. So over here we're showing kind of two substrate molecules represented by these two half circles. They're entering into the active site of our enzyme. And what we see in the next step is it almost looks like our enzyme is hugging our substrate molecule. It almost looks like there's two little hands right here, right, that are coming out and kind of wrapping around our substrate molecule. And this is what we call induced fit. So the enzyme is changing its shape slightly to create a very snug fit with our substrate. And this creates a nice little environment for the chemical reaction to proceed. And what is released from our active site is our product. So you see the two substrates have been joined together to make our product. And then our enzyme is ready to go again, right? We know that the enzymes are not consumed by the reaction at all. They're not changed by it. They're ready to go and accept new substrate molecules. So I always think of an enzyme as like a judge. A judge can bring two people together in matrimony, right? So bringing two individuals together, and then they go off in wedded bliss, and that judge is now ready to perform another wedding ceremony, right? They can take two more individuals, perform the marriage ceremony, and the two of them go off as a couple. Now, most enzymes can perform the reverse process as well. So we could take that married couple and sign off on a divorce decree, right? The enzyme could be the judge that signs off on a divorce decree, and the two go off as separate individuals. Okay, so a lot of times enzymes catalyze the forward and the reverse reactions. But keep in mind that that active site has to will only accommodate certain substrates, right? So the shape of the active site has to match the shape of the substrate. So there's certain enzymes that will catalyze certain reactions, and they won't catalyze something very similar. So I mentioned sucrase as the enzyme that could speed up our reaction by breaking down that sucrose molecule, our sugar cube. But it won't work if we try maltose, a very similar disaccharide. Maltose cannot enter into the active site of a sucrase enzyme. Okay, so they're very specific as far as the chemical reactions that they help, or the substrates that can come in and bind within the active site of our enzyme. So this is how they help to catalyze reactions. They help to create a nice little environment, a nice little microenvironment where the chemical reaction can proceed. So let's look at an example here. So this is asking you to identify the substrate, identify the product, and identify the enzyme. So remember, on the left-hand side of our arrow will be our substrate molecules. On the right-hand side will be our products. So sucrose is our substrate that is going to fit into the active site of our enzyme, which is sucrase, right? You recognize the enzyme because it has an ASE ending on it. Sucrase will help to bring about the conversion of sucrose into the products of glucose and fructose, which will then leave the active site. The enzyme sucrase can then go back, accept a new sucrose molecule, and convert them to glucose and fructose. Now, there are a few things that can affect how the enzyme does its job. So temperature and pH are two factors that can affect the activity of the enzymes. So if you think about it, if you increase the temperature, just like I said with the water and the sugar cube, if I heat up the water, then the sugar cube will dissolve. And that's because these molecules are bouncing around from all that thermal energy and coming in contact with each other, and they'll break apart. So that happens up to a point. So in our graph over here for temperature, we see as temperature increases, the rate of the reaction increases. But then you see our curve then falls off as temperature increases a little bit more. And what happens is, for these enzymes, the temperature becomes too much. It's almost like the thermal agitation is too much, and the bonds that are holding the enzyme molecule fall apart. They break. It's been agitated too much so that the bonds break. And when the bonds of the enzyme molecule break, the enzyme loses its shape. There's no more active site for a substrate to bind to, and the enzyme cannot function. When an enzyme loses its shape, we say that it has denatured. Remember, we said structure determines function. If it loses its structure, it cannot function. Okay? Same thing with pH. So most enzymes have an optimal pH at which they can function. After that, you see, so once again, pH increasing from 0 up to 10, right? So we have, in this case, some very acidic enzymes that work great in your stomach at a low pH, but as the pH becomes more towards the neutral, the activity of enzyme drops off. We have other enzymes that work in the basic environment, but they're not going to work under acidic conditions. So the same thing happens where the enzymes denature. They have an optimal pH at which they function. So think about, like, if you've ever left milk in your refrigerator for too long, past the expiration date, and you like swirl your milk around and you hear these like little chunks in the bottom of your milk. What has happened is that the protein that's in your milk has denatured out of solution and has become a solid. And that's because the pH of the milk has changed over time as the milk has kind of broken down. And so it's produced kind of more of an acid, and it's become more acidic, and the protein that is in your milk has denatured. Now, I'm not saying it's an enzyme, but enzymes are proteins, so it's kind of the same thing how it works. Also, if you think about when you cook an egg, right? You drop an egg into a frying pan, you see the egg white, and as you cook it, that clear portion of the egg turns white. So the protein in that clear portion has denatured due to the high temperature when you're frying up your egg, and it has precipitated out of solution and turned white. So it's the same idea with the enzymes. When the temperature gets too high, they're going to lose their shape and they're not going to function. Okay?